And amazingly, everything works beautifully, like always. And it is, Robin, can you hear me?
Yes, I can.
Excellent. Okay, so Robin is online and we are there, and it's a great pleasure to have him here. It's actually, which session was, I think it was, who said that he's my hero? Diana. And Robin is actually one of my heroes, I have to say that, because I studied a lot of effective population size. I think he once rejected one of my papers. I can't guarantee that, but I think it was him. But we finally got it through. So I had a long hidden history with him. He probably won't know, but he's actually one who thought probably the most of in the world about effective population size. And it's a really great pleasure to have him here today. And I just show my camera, so because you can see me almost crying. No, it's too much. And I think it's enough for an intro. He is Mr. Effective Population Size, has published, helped me, hundreds of papers on that issue. And it's really great to have him here. So please make him welcome if you can. And I hand over to you, Robin. Thanks a lot.
Well, thanks very much, Bernd, for those kind words. I got to say, several times I've sworn this is the last damn paper I'm ever going to write about NE. But it never seems to hold, because every time you turn around, some new wrinkle comes up that needs investigating. So that's why I keep publishing about it. I'm going to try to share my screen now so you can look. Let's see. Are you seeing the PowerPoint now?
No, it hasn't come up yet.
Okay. So let's see, Chrome tab.
In the center of there's this arrow, the up arrow. You click there, and then it asks you to select a window.
Yeah, I've done that. It worked when I was in
the backstage.
Yeah, well, it worked when I was in Firefox. Now it's got options are Chrome tab, window, or entire screen.
Yeah, you can. That's exactly what you want. But if that's if it doesn't work, what I can do, I have the slides you sent yesterday here. I can share my screen, and then you just tell me to click through.
Yeah, let's see.
So when you have entire
Chrome tab and then click my.
You can share the entire screen, and then yeah.
All right, maybe I'll share the entire screen.
Yeah.
But nothing happens when I try to click that.
Yes, so you have to select entire screen, and then you have to choose the icon of the screen. You can see it at the left. So there are
I got three options: Chrome tab, window, and entire screen.
Okay, go to entire screen, and then when you click,
nothing happens. So that doesn't seem to be a viable option.
Yes. So you have to select again the screen. There is you need to select the screen in that tab. You need to select the screen again.
Okay, so I will cancel this. Present to the audience, share screen. Chrome tab, window, entire screen. Okay, now it says I can do entire screen. So now if I select, now can you see?
No yet.
Not yet. Well, I'm sharing the entire screen, and my entire screen is my PowerPoint presentation. So
Okay.
I'm not sure what else I should be doing.
Okay, maybe we can go with the plan of Bernd. He can open the PowerPoint and you can say, go to the next slide.
Yeah. Okay.
Can you
All
share,
right.
Bernd?
Yeah. So Bernd wants to just show my slides from yesterday that I sent.
Can you see the slides now, Robin?
Yes, I can. That's good.
Okay.
So I'd like to take questions if there's specific questions that come up as we're going. I'm happy to take them if somebody's going to monitor the chat questions.
Yes, I will monitor the questions and let you know.
Okay, good. Okay, next slide. So this is a real brief roadmap for today. I'm going to talk about understanding and estimating effective size, both from demographics and genetic standpoint. And then I'll take a few minutes at the end talking about some simple R code that you have access to that just explores some of the principles I'm talking about. I'm not going to go through that in detail because the actual hands-on exercises today in the second hour are going to be led by Bernd and Luis. Okay, next slide. So the impetus incentive idea for my title came from this self-published manual of Volkswagen repair that I had a bunch of Volkswagen Beetles way back in my youth that I used this book to help keep them alive. And so this motivated me to prepare this paper I published last year. Next slide. It's basically a self-help manual for understanding effective population size. And it's a bit like a car repair because the whole concept's very elegant and simple, but as soon as you open the hood and start tinkering around in the workings, things can get very complicated. And my colleague Kathleen did this knockoff of the cover of the auto repair manual. Okay, next slide. We were asked or encouraged to sprinkle things related to Star Wars in our talks. And I got to say, I'm not really a Star Wars geek. However, there is one useful or perhaps interesting factoid here, because the first Star Wars movie came out in the summer of 1977, and that was the first real date I had with my future wife. Next slide. It didn't turn out so well. It turns out that she really has essentially zero interest in science fiction, and it seems to run in the family too. Nevertheless, we did get married and we're still married. So that's all I'm going to say really about Star Wars. Okay, next slide. So the very basics about effective population size is to remember it's just a number. And it's analogous to the census size, but it's generally a different number, and usually it's smaller. And the census size encapsulates information about the demographic consequences of population size, things like competition, predation, birth and death rates, and so on. Also, things like how fast a epidemic spreads through a population, that depends on the census size. But the evolutionary consequences of population size, things like rate of genetic drift or loss of genetic variability, those all depend on affected population size. And so to really understand what's going on in populations, particularly for applied conservation and management, you really need to understand both numbers and how they're related. Okay, next slide. First thing to note about affected population size is that it is the relationship between NE and genetic drift is very nonlinear. So all these evolutionary processes associated with genetic drift, their rate is inversely proportional to genetic drift. So the point about that, there's 2 points, take-home messages from that fact. One is, once NE gets pretty big, say 500 or 1,000, 1 over NE is really small. There isn't much genetic drift. And it doesn't matter that much how much bigger NE gets, because genetic drift is already really small. But once it gets fairly small, say below 100 or definitely in the low double digits, the rate of increase starts to increase very rapidly. So as you move to the left on this x-axis, the rate of change due to drift goes up very fast. That's why conservation biology and conservation genetics are particularly interested in low effective sizes. How do you keep things from getting too low? Okay, next slide. There are 4 evolutionary forces that are responsible for genetic diversity and how it changes over time. And genetic drift, or effective population size, is the determinant of genetic drift. Next slide. It just highlights genetic drift, I think. But in general, most cases, all 4 of these forces could be acting on a population. So genetic drift interacts with these other forces to produce the total changes in genetic variation that we see in real populations. Next slide. There's a big question. How big does NE have to be? Most or all of you maybe have heard of the 5,500 rule. It's not a rule like gravity is a biological or physical law. It's just really a rule of thumb. And the 50 number comes from empirical data for human breeding of livestock, pigeons, things like that. And they found that inbreeding at a level of about 1% tends to be tolerated, at least in the short term. So and that's equal to change in inbreeding is 1 over twice effective size. So that's the basis for the 50 number. It's good not to get below there in the short term. 500 came from some data on Drosophila, where they had some evidence that the effective size of 500 was large enough that mutations that created actual physical traits would be, you'd have enough mutation to keep a balance between new mutations being added and loss of additive genetic variation by genetic drift. So 500 is supposed to be about a threshold for long-term sustainability. I'm not going to talk about the mutational meltdown idea, which is much higher, but generally it really requires totally isolated populations to be meaningful. And all this really applies to closed populations, but particularly for that mutational meltdown number. Next slide. So recently, Dick Frankham and colleagues think that it should be revised upwards to 100, 1,000. Not everybody agrees, and I don't think there's really a consensus, but just remember that these are just rules of thumb. So somewhere in that range are probably important transitions. Next slide, please. There are some simple demographic formulas that describe how effective size works in terms of demographic parameters. A lot of interest in the NE to N ratio. So how small is NE compared to N? And a very good approximation to this is this simple formula where the denominator is the ratio of the variance in offspring number to the mean. And K is just a vector of the number of offspring produced by each parent. And so you can see that the ratio there is really important, and ratio of variance to mean ratio really is what determines the NE to N ratio. Next slide. And if the ratio, and go ahead to the next slide too. If that ratio is 1, meaning the variance equals the mean, that's what occurs in a Poisson distribution, which is pretty close to random mating. So if you have what's called a Wright-Fisher random mating population, it's going to behave pretty much like a situation where effective size equals the census size. But at the, in general, the variance is going to be greater than the mean in real populations. So how much greater than the mean it is going to depend how, determine really how small the NE to N ratio gets. Next slide. There's different definitions, way to define or name NE in literature. For your purposes, the two most relevant ones are the variance NE, which is defined in terms of rate of allele frequency change, inbreeding NE, which is defined in terms of the rate of increase in inbreeding. And for most practical purposes, and why is explained in some of my recent papers, I think inbreeding NE is generally much more useful. It applies to the parents. So if you get an estimate of inbreeding NE, you can think about it in terms of the parents that produced your sample. There's a handy formula that doesn't require you to know the actual total numbers of individuals. This is in one of our papers, but it just requires that vector of offspring numbers to calculate. We'll look at that a little later. Next slide. For me, the best way to think about effective size is in terms of a lottery. That's a special kind of lottery. The winners are the ones who get to produce offspring in the next generation. And so you have a separate draw of the lottery for each offspring to pick the parents. If it's diploid, you have to have 2 draws to get the 2 parents. And if you have a Wright-Fisher ideal population, then everybody has equal chance in the lottery. So they all have the same number of tickets in the fishbowl, and the drawing is fair. And furthermore, whoever is doing the drawing has to put the names back after they pick the winner. So whoever got to father or mother the first offspring has no effect on who gets chosen next time. That's exactly how the Wright-Fisher population works. Next slide, please. But what happens in the real world is, in general, the lotteries aren't fair. Not everybody has the same number of tickets. So they're, before you even start the lottery, they're expected winners and losers. And that is what stratifies this population into people with different expectations of reproductive success. That's what reduces NE compared to N. So we'll look at a few things that can cause that. Next. Well, the first is separate sexes. If you have males and females, you have to have 2 lotteries. So you have to have one poll for the male names and one for the female names. And unless the numbers of each sex are equal, whichever is the less numerous sex, those people on average, those parents on average are going to get picked more often. If there's 50 males and 100 females, on average, every male is going to get picked twice as much as every average female. That creates disparities among the sexes in mean reproductive success and reduces NE. But it's not a very strong effect. That's Wright's famous formula there, which can be expressed in terms of the NE to N ratio. Next slide. It just shows that, I think, yeah, even if you have a 60-40 sex ratio split, which is fairly skewed, it only reduces NE to N by a few percent. So you have to have a pretty dramatic sex ratio difference to have a big effect on NE. Next slide. This is the thing that reduces effective size the most. Individual differences. Some individuals are just bigger, stronger, faster, smarter, sexier, whatever. They have more tickets in the lottery, so they're likely to produce more offspring even before you start the lottery. And this can occur for genetic reasons or non-genetic reasons. Some cute title a few years ago called this pluck or luck. Okay, next slide. There's also stratification due to age structure. If you have heteroparaspecies, and most species don't have discrete generations, most species are heteroparous, effective size depends on the lifetime variation in reproductive success. So with overlapping generations, some individuals are more equal than others. First, because they survive. That means they get to enter more lotteries, a new lottery every year, say, and often fecundity increases with age. So next slide. Here's some empirical data for a bunch of marine species, all that show relative fecundity increases roughly linear with age in these. SBT is southern bluefin tuna, which is a big deal down there in Australia. Next slide. It's not just marine or aquatic species. These are some data from colleagues I worked with on black bears in Michigan. And fecundity increases with age in both sexes there. So what happens then is individuals that live longer get a double bonus. They get to enter more lotteries, and in later years, the lotteries, they have relatively more tickets. So that really helps increase the disparity in lifetime reproductive success among individuals that do and do not live a long time. Next slide. With age structure, you have to think about two effective sizes. NB has been used to refer to the effective number of breeders per year. It's basically the single generation formula for effective population size applied to all the breeders in 1 year or season. But effective size per generation, the best model is by Bill Hill. Next slide. And his formula, you can see it has T is generation length, that's in the numerator, and the denominator is this VK star or asterisk, which is lifetime variance in reproductive success. So you can see the bigger that gets, the smaller effective size gets. Next slide. Yeah, I just said that. Okay, next slide. For heteroparous species, other factors that affect the ratio of NE to N are skip breeding. That's where individuals, particularly females, may not reproduce every year. This actually tends to increase NE per generation. because it keeps some females from dominating reproduction every single year. Persistent individual differences, these don't affect yearly effective size, but they can have a big effect in reducing effective size per generation because they increase lifetime variance in offspring number. These are situations where some individuals are consistently above average or below average at producing offspring year after year after year. And there are lots of reasons why that might be the case. Small litter size, on the other hand, tends to increase both effective sizes because it constrains how much variation, at least the female, how large the family size can be for females. Again, it just puts a limit on how extreme that reproductive success can be. Next slide. Another big factor that affects the effective size is migration and spatial structure. So this is a huge topic. I'm not really going to go into much detail here, except to point out it's in accounting for migration, it's really important to distinguish between local effective size and metapopulation or global effective size. And this is particularly true, as we'll see, if you're estimating historical NE. It's really crucial to consider the effects of gene flow. Next slide. Now, Dennis Hedcock came up several decades ago with an interesting idea about that's been called sweepstakes reproductive success. His idea was that, at least in the marine realm, often it may be the case that you don't have individual success of individuals just by flipping a coin, see if they survive or not. His idea was that often entire families, say all the eggs produced by a female, will either live or die as a unit. And that could be because they never float to a place in the ocean where there's any food. They get all gobbled up by a predator when they're all in one mass. So this would, if you want to do this in a lottery, you have to have a different kind. In a Wright Fisher or standard kind of lottery, each draw picks a parent of one offspring. But to model sweepstake reproductive success, you'd have to have a situation where each draw, you pick the parents that get to produce many offspring, say 10. Okay, whoever's names I draw out of the hat right now are allowed to produce 10 offspring, or 100, or 10 million. So there's really no limit to how high that could be. And that sort of scenario is really the only way you can get these tiny NE to N ratios that have been proposed for a bunch of marine species. Next slide. So that's the demographic part. This second half of my talk, I'm going to talk about genetic estimates of effective size, which I think is more oriented toward what people are thinking about for this conference. But the demographic background is really important. So that's why we went into it. So this is the same graph I showed you earlier, relationship between effective size and rate of genetic change. But at some point, somebody realized that, well, maybe instead of taking effective size and estimating the rate of change or predicting the rate of change, you can work backwards. If you could measure or estimate some rate of genetic change, you might be able to estimate effective size. So that's the whole basis for this, for all of these methods that try to estimate effective size. They're based on some relationship like this, and you measure or estimate some genetic index. And this was initially used with the temporal method over 50 years ago. Oops. Maybe we can reset there. And yeah, we're about halfway through, I think.
Yeah, sorry, my mistake.
It's okay. Yeah, that.
Come on. All right, let's start from there.
Yeah, Yeah, that's the one right there in the middle. Click. Okay. So if you go next slide from there. So genetic methods can provide information about historical NE. We'll talk a little bit about that, and I think Bernd and Luis will. Contemporary NE, which is basically NE spanned by the time of your samples, and also bottlenecks. So I'll talk a little bit about each of these. Next slide. Next slide. Yeah. Long-term NE, so this is an equilibrium idea. It's based on the idea that if you have a population that's been in long-term equilibrium between genetic drift and all the other evolutionary forces, then there should be a predictable relationship between NE and the amount of, say, heterozygosity or gene diversity you have. So they're based on these simple equations you've probably seen here, and from them you can get a predictor. If you have to measure something like heterozygosity, and you have to be able to estimate the mutation rate, and you have to assume that populations at equilibrium, migration, mutation, drift equilibrium, which most populations aren't. And you have to believe that everything you're assaying genetic markers is absolutely 100% neutral. That's why I've never been a big fan of these estimates, because there's so many things that have to be met. Okay, so that's all I'm going to say about the long-term equilibrium estimates. Next slide. Reconstructing changes in historical NE has become a big deal recently. There have been methods available to do this for at least 20 years or so, but the new method has been developed rapidly in the last decade or so. This is a very nice review that tries to put a lot of this stuff in context you probably should look at. And you're going to hear more about it in the hands-on, I think. Next slide. Okay, yeah, so here again, just a caveat. These things may, many of these methods will not try to estimate a long-term historic NE or just a contemporary NE, but they will try to estimate effective size at different time points in the past. The key thing to remember about, I believe, all of these methods is that even really very low gene flow pushes historical estimates more toward metapopulation or global NE, whereas we know from methods to estimate contemporary NE, they estimate mostly local NE unless you have pretty strong migration. So what this means is, even if you have a species where there's been no change in migration, say for the entire duration of millions of years in the past, if you apply some of these methods, and this population is part of a metapopulation, the part that's estimating recent NE is going to give you a signal of local NE, and the part of these methods that estimates historical NE is going to give you a signal mostly of metapopulation or maybe even species-wide NE. And it can look like a false signal of a bottleneck. So that's a real important potential problem for these methods. Okay, just flagging that caveat. Next slide. Bottleneck tests, they all have the same basic logic. And the logic is that equilibrium, there should be some sort of predictable relationship between two different genetic measures. In this case, the number of alleles and heterozygosity, there's a predictable relationship between those two indices at equilibrium. But if you have a bottleneck, the number of alleles just goes down faster because rare alleles are the first thing to go, and rare alleles have little effect on heterozygosity. So heterozygosity has a lot of inertia. So you get this disparity again after bottleneck. And so all of the bottleneck tests, they compare different things, but the same sort of logic. Next slide. Yeah, and this one just talks about the Garzon-Williamson test using microstats, same idea. Next slide. Methods that estimate contemporary NE, they're really two kinds, two samples, the temporal change in allele frequency. The temporal method was the first one. And you can also measure changes in genetic diversity, like loss heterozygosity. But that doesn't have as much power. So the other ones that are widely used are two one-sample methods based on LD and CPGIPs. Next slide. Just about all of these methods have similar assumptions, and the assumptions have been evaluated in many cases, but in lots of different papers published in lots of journals over many years. So in my Idiot's Guide paper, I have a table that tries to summarize most of the evaluations that have been done. But two that are particularly important, most species don't have discrete generations. And also I'm going to talk about the fact that, particularly with genomic scale data sets, loci or locus pairs are not independent. So that affects precision. Next slide. Temporal estimates, pretty simple concept. You take a sample at one point, take another sample, same population. You compare allele frequencies. This index of allele frequency change, temporal F, is closely related FST. And there is a predictable relationship between that and effective size. That's the basis for the temporal method. Next slide. The LD method is based on non-random associations of alleles at different loci. R squared, the correlation coefficient, is a measure of LD. The squared correlation coefficient, that removes the sign. So if you're dealing with genetic drift, the sign of the LD doesn't matter. It's just the magnitude. Next slide. So for the LD method, if the loci are not physically linked, say if they're on different chromosomes, and if there's no selection or migration, there's this simple expectation approximate between the expectation of the value of R squared, which has to be range from 0 to 1, is a function of two things. Next slide. The first part, the 1/3 NE, is a drift signal. Next slide. But you also have random noise that's a function of how large your sample is. And you can see 1/S, even if S and NE are the same, the 1/S is larger. That term is larger than the drift signal. So it's a big deal. And that's why the LD method only works if you get lots and lots of data to average. Next slide. So the way you do these estimates, same thing with the temporal method, you have an expected amount of noise contributed by sampling error. You subtract that from the raw statistic. And what's left in theory should be due only to genetic drift. So that's why the -1/S is there in the denominator. And that's how you get an estimate. Now, it turns out this estimator is biased, and so I was involved with some others that developed ways to correct this bias. That's what LDNE, for example, does. It's a bias-corrected version of Hill's method. Next slide. Oh, and with linkage, well, if you're talking about the loci on the same chromosome, R squared also depends on the recombination fraction. We'll talk about that in a minute. Next slide. So this just shows a little bit about precision. So this two scenarios, different NEs, order of magnitude apart, both had like 20 kind of decent microsat loci. If true NE is 100, you can see all the estimates with the LDNE method were really close to 100. So that's a strong signal. It's pretty easy to detect. If NE is 1,000, remember 1 over NE is the signal. It's a lot more diffuse. It's smaller and harder to pin down. So with the same amount of data, you don't get as tight a window. Next slide. So why is when NE is large, why is it so hard to estimate? And then the basic reason is the drift signal gets really small. Next slide. Yeah, this just shows two scenarios where NE is 100 with the temporal method. You can see in that scenario, the drift signal is pretty darn big. So if you got enough data, you should be able to nail that down pretty well. But if same amount of data, NE is 10,000, the drift signal is tiny. It's like a needle in a haystack compared to the noise from sampling error. So that's, in a nutshell, why it's really hard when the drift signal is small, particularly compared to sampling error. Next slide. And there's only a certain amount you can do by sampling more and more loci to compensate for that. Now, this lack of independence, with lots of loci, some pairs are inevitably linked or in the same chromosome. That's because you get genomic scale data sets. The average chromosome number for even vertebrates is like 25. So you're cramming millions of loci into 25 chromosomes. You know, by the millionth locus, there is no place you can put it. It isn't really, really close to at least one locus already in your data set. So what happens for the LD method? This can bias estimates if you're assuming the loci are unlinked. That's number one. It also creates lack of independence because the loci are inherited as a unit, and so you don't have as much information as you think if you treat them all as independent. Next slide. So for dealing with linkage and you want to estimate effective size, if you want to estimate contemporary NE, you could restrict comparisons to loci in different chromosomes. If you have the data, that's easy. That's an option in any estimator, just restrict to the comparisons. You could sort pairs of loci based on the recombination fraction. And the advantage of doing that is if you use loci in the same chromosome, particularly ones that are tightly linked, they can provide information about NE in the past. And so that's one of the methods that allows people to get a window in historical patterns of NE. Gone as a recent method that does that. The third thing you can do is adjust for the expected bias, again, if you want to estimate contemporary NE. And the next slide shows one way we try to do that. Oh, no, that's the next slide. This shows the method. It's basically the same kind of method used in Gone, but it's an earlier version. So this basically takes the pairs of loci. You have to have detailed linkage map data. You put the pairs of loci in different bins, and each bin of how low the recombination fraction is will tell you which time period your estimate applies to. So this has been used to estimate effective size in different populations back quite a few hundreds or maybe thousands of generations. Next slide. Here's something that illustrates why pruning LD, which is kind of a thing a lot of people like to do, is only marginally effective, particularly if you're dealing with effective sites. You can see in the top panel, this shows a situation where you have the full distribution of R-squared values that mix both comparisons across loci and within loci. And in the bottom panel, they're separated. So you see the light gray is different chromosome comparisons, and the dark ones are the same. What you can see is if you decide to prune, I want to get rid of all the R-squared values on different chromosomes. Well, you could prune to about 0.4 or something here pretty safely, and you wouldn't get rid of many comparisons from the same chromosome. But if you go lower than that, you got a mix of any R-squared value you can pick. You can get both cross-chromosome pairs and within chromosome pairs producing the same R-squared values. So it's not a reliable way of distinguishing between linkage and not. Next slide. So in this paper my son led back in 2016, we looked at what happens if you just use all the loci. And it turns out the bias, you'll get downward bias because linkage due to LD, not due to drift, but due to physical linkage, is being mistaken for LD due to drift. Turns out the bias is a simple function of NA. The simple function of the number of chromosomes, which is a log scale on the x-axis. So that allowed us to produce a simple linear adjustment. It seems to work pretty well in a pretty wide range of scenarios. If you don't have detailed information to exclude the within locus comparisons, you could do that. Next slide. How about this lack of independence precision? In economic field, there's this well-defined phenomenon, which is called diminishing marginal utility. And basic idea is sort of after you get 2 or 3 or 5 TV sets in your house, the 6th TV set, which you probably have to put in a bathroom or a closet somewhere, it doesn't give you much more benefit. The first TV set would be the biggest benefit. Or here in this case, it's on a hot day, how much joy you get from the first beer. So the point is, the same thing applies to loci. If you have more and more loci at some point in genomic scale data sets, you're just getting the same information again because you've already extracted as much information as you can from the data set. So where do those thresholds apply? It turns out to be a hard problem. Next slide. We looked at a few years ago, and we wanted to see, well, for the LD method and FST, which is really analogous to the temporal method. These are theoretical CIs, confidence intervals around a point estimate where true NE, say in the top one was 200. If you assumed, if you had, let's say, 1,000 or 10,000 loci SNPs and did all pairwise comparisons, well, you'd have 500 million pairwise comparisons on the 10 to the 4th SNPs. And if those were all totally independent information, your CI for your point estimate would be like 200 plus or minus 0.05 or something, ridiculously small. And you can usefully use more SNPs for FST, but still you get a point where in theory you would basically know the answer. But we know that's not the case because these are not independent. How how much less information do you have? Let's go to the next slide. So we did a brute force paper where we simulated lots of data sets with known NE and known numbers of loci and known numbers of chromosomes. And we had to be very careful to control extraneous sources of variation because we're studying the empirical variance of your estimate as you added more and more loci. And the variances get really small, so you had to do lots of replication. But the bottom line is that the actual true confidence intervals are much wider. And that's particularly true for the LD method than you would get if you just assume everything is totally independent. And particularly for the LD method, once you get about 1,000 random loci, you're not getting very much new information from additional pairs. You can profitably use at least 10,000 or maybe 100,000 loci for individual things like FST or temporal F. But still, you don't get as much precision as you would think if you assume everything is independent. Okay, next slide. So one of the consequences of this, if you think about experimental design, it really runs counter to what has kind of been happening in the last 2 decades, is the cost of DNA sequencing has dropped by many orders of magnitude, just precipitously. But the cost of doing field work has not. If anything, it's gone up. So what this has caused people to want to do is to say, well, I'll compensate. I can only go out and get 6 individuals, but I'll do whole genome sequencing, and then I'll know everything about those 6 individuals. Well, in a sense you will, but you won't know everything about the population because there are 2 major sources of uncertainty in any sort of empirical data. There's uncertainty associated with sampling genes in the individuals you have. And there's uncertainty associated with sampling individuals from the population as a whole. And to only some extent can you trade off one with the other. So really, as the last slide should have made clear, there is a limit to how much you can compensate for having small numbers of individuals by just cranking up the number of loci you're going to look at, because you will max out relatively quickly the useful information you can extract. Now, that doesn't mean that whole genome sequencing, for example, doesn't have very useful things. There are other reasons besides just mass increasing the number of markers that you could get from detailed link, detailed map distance, different kinds of loci, everything. But I'm just talking about the brute force kind of increasing the number of markers you have. There are some limits to how much that's going to help you. So just remember, individuals really are really valuable in these sort of experimental designs. Next slide. Now, in the back of the Idiot's Guide is a thing. These are things I've heard people contact me all the time with questions. And a lot of these things are answered in various papers, but I realized, well, it'd be good to put them all in one place. I'm not going to go into them now, but please have a look. The explanations for why they're not generally a good idea is in the paper. Next slide. So this is where I was going to just really quickly run through one chunk of my R code before turning it over, but we got a little late start, so I think I may skip this. So what I put together is some R code. I did it initially for a different project, and then I modified it for this and other things. It's bunch of R code. I don't know how many lines, a few hundred lines, but basically it starts out by implementing simple Wright-Fisher modeling populations. It's in R. It's very easy to exactly mimic the lottery draw I talked about for picking the parents. And you can see the sort of consequences of different sex ratios. You can also see consequences of different individuals, different probabilities of producing offspring. And then later, the code adds genetics. And so you can track estimates, loss of heterozygosity, and things like that. But it's pretty well commented. And Luis and Bernd turned my code into a nice R markdown thing that's available to you when you get, I don't know if it's in the GitHub.
Yeah, it's in the comment there. In the today's tutorial at the end, it's there, so they're all
right.
You can run them, and I'm happy to show them when I go through my exercises.
Okay, if you want to show any of it, that's fine, but don't feel a need to, because I think it's mostly self-explanatory. It would be good to get feedback. If anybody does end up using it, you can take it home with you, play around with it. If you have any feedback, it'd be good to hear that. So I think I'll stop there. And I'm not sure if any questions popped up, but happy to take any here. Or I will hang out at the next session and see how that goes too.
Although that's scary. So you see all the things what I do when I say I estimated from genetics data, and you just told me I shouldn't. That's excellent. Are there any questions for Robin at the moment? So please put them into the Q&A. And actually there are some, aren't there? Yeah, okay. Is that, yeah, okay. Can I put them on stage and you want to answer them, Robin?
Sure. Well, there's quite a few methods specifically designed for structured populations. There's, in terms of estimating effective size, there is a model by Wang and Whitlock about 20 years ago that uses the temporal method and will give you an estimate of effective size and also an estimate of migration rate. But it's only applicable to a very specific scenario where you have a local population that gets constant immigration from a infinitely large source population. So it's sort of like a mainland island population. So it's kind of an odd scenario, but there is that one. And there are quite a few papers that you can see referenced in my Idiot's Guide table that have looked at effects of migration on effective size estimates.
Another one from Peter.
Yeah. What sample? Well, it's easy to say this, but obviously as large as possible. And if you want to estimate effective size, anything really less than 20 is really pushing it. Unless if your effective size is also, say, less than 100, then that may be adequate. Because then the drift signal is going to be really strong. And your 20 might be some reasonable fraction of the population. But if you got a population that may have an effective size in many hundreds or thousands, and you're trying to estimate something about that with just 20 individuals, the results are going to depend a lot on which 20 individuals you happen to pick or happen to get in your sample. And if you happen to have two siblings, say, in your sample, but it's really from a large population and they just got sampled together because sampling randomly is almost impossible in the real world, that could have a really big effect. So it's not just the small size of the sample, but it's also, because it's small, these weird departures from randomness can have a relatively really big effect. There is one situation where it can help, though. If the biggest requirement for large sample sizes occurs when you want to estimate, say, contemporary evolutionary processes. That's what I focus most on. If you want to estimate something in the deep time with coalescent methods, then in a lot of those scenarios, a fairly small number of individuals can actually be adequate. And the reason is because their genetic traits, their genetic characteristics just coalesce in time farther back you go. And so they pretty much end up leading to the same answer, even if they might really distort an analysis that's really focusing on contemporary processes. So that is a difference. Yeah, excellent question. There is, I just had Armando Caballero just sent me a manuscript of his to review, and it's a review paper, and in that, it's not published yet, I don't think, but it cited another recent paper by Santiago that tried to do exactly that for the GONE methods. They had known about this issue, and they'd even talked about it in some of their papers, but look for a paper by Santiago et al. It'd be either 2024 or 2025 in some high-profile journal that does attempt to do exactly that, at least for the GONE method. And there may be ways to try to do it for other methods, but I'm not sure about that. Okay, so I guess you want to estimate, are you talking about estimating, historic or contemporary? I would, for a population that's, drift with 500 or 1,000 or so, you're at the situation where genetic drift is relatively small, the drift signal's weak. Ideally, I'd want to have at least 30 to 50 individuals for that. There's no magic number, black and white, yes or no.
Robin, there are some more questions. Are you happy to go through some more, or you?
No, I'm happy, but it's eating into your time too. So.
Yeah, no, that's totally fine. I think it's, the tutorial is there and it will be there for the next whatever. So
Okay.
people can go through, and that's totally fine. I think it's more valuable to ask you while you're on stage.
No, it's better because, I mean, when you give a talk for an hour and nobody asks any questions, it's kind of discouraging. But so I think it's good to have a question. Okay, let's see. I think I may need to, oh yeah, let's scroll down. Some of these questions are a little longer. Okay.
Can you see it? I can read it.
I know I can see it. It's just I had to make my font smaller so I could read more of the question without scrolling down. Oh, I see. Okay, so if you have decrease in breeders, so yeah, this goes back to one of my slides. So if you have skip breeding, let's say, I think this question may relate to that. And so let's say 20% of the females don't breed this year because they used all their energy the year before migrating as sea turtles or something to go on a beach. So that's why NB would be reduced if you have skip breeding, because you'd have fewer females, you may have a skewed sex ratio then, or there'd be a change of their different parental investment. So females invest more than males. Let's see. Yeah. So, well, let's see. Different parental investment. You mean different parental investment to how big the gametes are? So yeah, certainly that's the case in most cases. Sperm isn't very hard to make. And a sea turtle, if we're talking about a sea turtle, that's a big parental investment. But still, the genetic consequences are going to be roughly comparable for the two sexes, I think. If you had 20% fewer males, it potentially had a similar genetic consequence for NB. Yeah.
I just have to find all the new ones. They're coming in more and more. And sorry if I skipped someone. I'm just overwhelmed at the moment.
Filtering SNP data. Yeah. I have to confess, for my PhD thesis, I did all my genetic data. I got all my samples, did all the lab work and everything, but that was the age of Alzheimer's. And I've never done any DNA data collection in the lab myself. So I do know enough about filtering SNP data to know that it's really an important step and a lot of decisions you have to make. Somebody has to make that can have a big effect on what data comes out. And the filtering decisions also can have a big effect on the analysis you do and the conclusions, including any estimation. But I am not an expert on this. But one of the things I think you definitely want to do, and I did a paper with Marty Cardos a year ago where we looked at some other published data on wolves that had relied on low coverage sequence data to estimate effective size. And there were some real problems there because there was a strong negative correlation, a strong positive correlation between read depth and estimated heterozygosity at the individual level. So that indicated that on average, the read depth was not sufficient to reliably call genotypes. And so a lot of them heterozygotes were being missed. And if that's the case, that's well known, say with the LD method, it can bias estimates of effective size, and it can have consequences for all sorts of other analyses. So that is one thing I would definitely look at. Look at read depth and things like missing data as a function of read depth and heterozygosity or genetic diversity as a function of read depth. Those would be really important first things to do. In addition to standard things like Hardy-Weinberg evaluations and so on. Yeah, I've heard this, and it may be affected. They have temperature sensitive sex determination, I think, and so could be affected by climate change. The females, my understanding, can control us to some extent by figuring out where in the beach or how deep they lay their eggs. But with 80 to 90% females demographically, that's generally would be considered good news for a population because there's, it's easier to get a high population growth rate. So yeah, 99%, 99 to 1 sex ratio can have a big effect on any. You can evaluate that pretty simply. The one formula I had, you can do it right here with 80%. It's 0.8 times 0.2 is 0.16 and multiply that by 4. So 0.16 times 4 is 0.64. So with 80-20 sex ratio, you expect the NE to N ratio to be reduced by about 1/3 just by the skewed sex ratio. So that's not a huge effect, but it's not trivial. And that would be in addition to everything else. But once you get below that, then you start to get some big effects. If you're talking 99%, that would have a big effect, reducing any order of magnitude or more.
Hey, sorry, there's some more questions. You're still happy to go? I'm happy to put them up, but
I'm okay. I'll need a beer pretty soon,
but give me a sec. I have to find which one hasn't been answered. This one is not. Yeah.
Yeah, great question. Yeah, sure. You can, it could be that you live longer just by luck. And so everything I talked about would apply even to individuals who live longer just by luck. They're more likely to have higher lifetime reproductive success. But if the reason you live long is at least partly because you have good genes, then the part of the effect here is due to selection, and its selection could be reason this individual is producing more offspring, and it could help improve the average fitness of the population over time. So it could be an adaptive strategy for the population. But it's also going to affect effective population size, which is what I was focusing on, because basically by definition, natural selection is differential reproduction in individuals with different genotypes. And the differential reproduction part of it is what increases the variance in reproductive success. So there is an interaction between selection. Strong selection creates stronger genetic drift because it creates disparity in offspring number among individuals. But, and this is kind of a conundrum, when you have lower effective size, selection isn't as effective or as efficient because genetic drift, the random noise of genetic drift becomes stronger, and selection has to be stronger to maintain the same effectiveness. It's the same reason casinos have to have really large volume if they have small margins in each of their gambling games. If they have huge volume, they can be sure of making a lot of money every day and every month. So that's an interesting question. There is this kind of odd feedback between selection and drift there.
Sorry, we torture you a little bit longer. It's totally fine. Hope so.
Individual behavior absolutely could affect any. Individual behavior could affect both the probability of surviving and the probability of reproducing. So it could go either way. I mean, if you're stupid, like some young males, go running around not knowing what they're doing, they're likely to die before they reach reproductive age. But if they become, if their behavior turned out to be attractive to females, then it could boost their reproductive output. And there could be trade-offs there too. So certainly it could and likely does affect. Okay. Thanks, Luciano. So we're talking about conservation management of threatened species. I think that in this case, the NE to N ratio is useful for two reasons. One is it's useful if you can estimate the NE, if you can use or use the yardstick, the NE to N ratio, and you are able to estimate N, it's a way to estimate NE, which maybe is what you're really interested in. So that's one way. But also the ratio itself, I think, is very informative, can be very informative about what's going on demographically in the population. So if you're able to get a reasonable estimate, say, of NE to N of a managed species, you can start thinking about some of the types of processes that I discussed earlier in the talk that might be going on to have produced that ratio. And if it's relatively high, you might think, oh, that's pretty neat. NE is small, but so is N. So we're doing a pretty good job of maximizing genetic diversity, given that we're stuck with just a small population. On the other hand, if your estimate of NE to N is, 0.05 or something, and you got a threatened species, there might be quite a bit of room for improving things by nudging some conditions that would try to minimize some of the factors that might reduce NE to N and promote some of the factors that might increase NE to N.
I have two more questions, I think, and if I've missed you, that's fine. Is that okay?
Yes. Yeah, sure. Yeah, I think, maybe not the same level of dependence. I think because if you have coalescent methods, like I said before, they're based on concept of a process that where basically the current samples of genetic traits in those are going to converge into a much smaller number of individuals as you work into the past. And so what happens there is that different individuals you might collect in the current time can tend to lead to more or less the same answer, more to a greater degree than they do if you're estimating something like effective population size of the parents that produce my sample. So yeah, so I think there is not quite as strict an issue with small sample sizes necessarily for a coalescent-based method. There's a nice paper by John Wakely and others several years ago that discusses this a bit.
Okay, I think that's the last one. I can wholeheartedly agree on that one. And now please send your kind emojis. We have no other choice to Robin. And thanks you a lot for your great presentation. You can see there's lots of interest in any, and maybe I can, because I have you there, I can basically grab you. You know, we have this beautiful tables where you can meet other people. And would it be okay if they wanted to take a bit of more time at some stage and we arrange an extra table with Robin somewhere, and then we have even more questions? Is that possible?
Yeah, sure. Yeah, that's fine.
Yeah.
Actually, you skipped, if you went to my last slide, I put my email up there in case people had questions about their own data or something. Yeah, I'd like to go. I was hoping to come when this is initially planned for an in-person meeting, because I lived in Australia back in the 70s and really had a great time. And I've been back several times, but it didn't work out this time to come, and it turned out not to be an in-person meeting anyway. But it would have been much more enjoyable. And I do like periodically to go to meetings like this where I actually get to meet young students, postdocs, and find out what they're working on, what they're thinking about. And getting questions like this is always useful to really see what people are worried about, interested in. So thanks a lot.
Okay, thanks a lot. We try maybe via email. We maybe present a date where we can have a meeting on the table, and if it works out, and it's really kind to provide your email. So please, everyone obviously has a chance to contact Robin if you wanted. Thanks a lot again, and give me a minute or two to get sorted, and then I will go into my tutorial. Thanks again to Robin. Okay. We have roughly half an hour, I guess, and I will go through the tutorial, obviously, and give me maybe some leeway if I mess something up. Today we have PopGen master session 7 to 9, so make sure that's the only one you should see, and we rightly go in there. Just to make sure I go in another session, but don't care about it. So fire up your Posit Cloud, and we try to go through that. And while it's loading, I probably can already start to share the idea of today's tutorial. Obviously, we do all the things which Robin was kind of warning us that we have to be careful. We try to estimate effective population size from genetic data. And it was really nice to see all the non-genetic versions, which I have to be acknowledging I have to do more. Because obviously that's some really nice thing you can do. The concept of the NE is obviously on the website, you can see. And there's some introduction, and we will do in our tutorial the contemporary NE based on linkage disequilibrium, which is the NE estimator method published by Robin and Doe, not sure what his first name is. And they use this, exploit this linkage disequilibrium relationship between effective population size and NE. Okay, let's go into it and see if that works. Okay, console and go to tutorial as before, and we should have tutorial number 7. I try to start that up as well, and you do as well. It will take a while, maybe. So I go back to here and explain a little bit the idea. Obviously, we will load the packages, and then we talk a little bit about filtering, because that's really important, as Robin just said. One of the main critical one is you want to have a certain minor allele frequency, because you don't want to get this noise, because in the end we estimate the linkage disequilibrium, and as Robin said in his formula, this was actually the better one. The effective population size is basically this estimate in inverse proportion. So if you get that wrong, even by just a fraction, this will affect your effective population size a lot. So we have to be careful, and therefore we need high quality SNP and as less noise as possible. So for example, here, in the real example, I show you the filtering later. Initially, I cheated a little bit, because what I did is very, where's my tutorial coming up? Try again. Give me a sec again. It doesn't like to be not here. Okay. So please start it up. This is a tutorial that runs quite a bit of code, and therefore it takes a while to come up. Hang on, where is it? There it is. Okay, great. So we go to part 1. Here's the introduction, as I said, and it gives you a little bit of overview. The first thing we do, we load some of the packages. Yep. So make sure you load them first. And then go here and download, which is important, the NE estimator package. And this is the function which I showed before, which downloads the NE estimator binary file from our package and puts it into the temporary folder, so we can run it directly from there. So if you execute the command, you should get something like this output. It's unzipped into the temporary folder. Okay, as I said a little bit, I cheat, because I will simulate some data, and the idea is the following. Obviously, in how good is the estimate and the simulation, and in a simulated way. Obviously, it should be pretty good. If the method doesn't work, if you simulate data, then you have trouble. And I come into a second set. When I say simulate, I use these commands. I will explain them more in the simulation session. This estimates, generates 50 individuals, 3,000 loci, and the effective population size is actually exactly that, because it's an ideal kind of scenario. And here, Robin told me that's wrong what I said here. I have to edit and to LD, because when you simulate the data, the individuals haven't mated, obviously. So you have to run it for some generation. That's what this code does. Basically, the simulation is artificial, and then we let them kind of reproduce for 5 generations in an ideal Wright-Fisher way. And therefore, we have kind of a population that is where the alleles are in Hardy-Weinberg, or loci in Hardy-Weinberg, and we have this linkage disequilibrium kind of effect in there as well, meaning we can now use our method, which is the any estimator method. Just to be aware, make you aware, any estimator is also has a GUI as well, so you can also run it from the GUI, and it has lots of settings. And I actually recommend it maybe once or twice to do that, because then you see all the settings which are there, and you may understand them better because there's also a nice documentation. Obviously, for convenience, it's nice to run it from R. So we can just do that by just copy paste it in there, and then hopefully that runs smoothly. It has to convert it first in a GenePop file, so it takes a while, and then it runs the any estimator, and you see a lot of output on the screen. And in the end, you should see what is that? A plot of effective population sizes based on the settings we gave here. And I go through the settings in a second. Nice thing you can see, we simulated 50 individuals, effective population should be 50, but even here you can see it's not, in an ideal case, it's not really coming up with exactly 50 individuals. It's biased, and depending on the different minor allele frequencies you filter here, these are singletons, and these are no filter, and this is filter for minor allele frequency of 0.05. You can set that with a critical value. You can filter for different minor allele frequencies, and then it runs it for different subsettings. The good news is, if you do that with your own data, and that's probably what I would encourage you to do in a second, if you run your own data and the filtering of the minor allele frequency doesn't affect too much your effective populations, maybe you have a good idea, this could be something, I'm onto it. Because as Robin pointed out, when you run it, you will find out it is very critical that you have really high quality SNPs, really high quality data. And as I said, in the end of this tutorial, we go through the filtering steps. At the moment, I just want to show how we run the settings. So I explained the critical version here. Singleton remove is another way to do it. It's basically you get away from loci which have only called once one allele, and you get rid of that. Yeah, 0 means no minor allele frequency filtering. 0.05 is 5%. Sometimes you want to filter for allele counts, and then I would recommend you filter it in dartR, and then run this with a 0 settings, because you have filtered it before to a certain amount, and you can run the critical equals 0, and it has the right settings because you filtered by hand before. What else do we have here in the function? We have mating session. This can be either random or, give me a sec, how we called it. As always, there's a help page, and you can hopefully see all the different settings. And oops, what's happening here? So you have mating, you have either random or it's called monogamy. If you have a species where you know that monogamous, you use a different kind of settings, because the formula Robin showed, the relationship changes depending on the mating system. Okay, maybe I give you a minute or two to catch up and see if it runs for you. And please show me in the chat, which I switched off, what's happening. And feel free to post some questions if you have some. Can I get some emojis if this runs for you? Okay, there are actual people out there. That's good. Okay, let's see. Yeah, as before, we don't know. You don't have any bad emojis. Maybe we can make that up. Maybe if you send me the wow emoji, I know it doesn't run. Okay. I couldn't see any. It's great. Good. So that's the way how you run that. But obviously there are lots of things to consider. As I said, minor allele frequency filter is one thing. Then there are a lot of other quality measurements you should do. You should get rid of missing data. You should make sure your read depth is high enough, so you're not missing, and you're not including loci which are of dubious quality. Okay. And what else can you filter? Individuals missing. Yeah, I think that's the main ones you do in terms of quality.
Where do we post a question?
In the chat.
In the chat, not the questions.
Yeah. Peter's asking now. Maybe post it in the Q&A session. Yeah. If you have any questions regarding at the moment the tutorial, if I come to it, please post them to the Q&A session. Okay. Can someone monitor that and make me aware of that? If there's some. Okay. Let's go through that. Let's look at the output. So the whole thing, when you look at this, is stored in an object called any result. And as you can see, there is an output, and it's kind of interesting to look at. This is the famous linkage disequilibrium value, and you can see you get an output for each of the settings. Remove singletons, no filter, and filter at 0.5. And then you get quite a few statistics. And those two statistics are the important ones. Why is that? Because this ratio, this value towards this value, is basically the effective population size. Okay, give me a sec with the question. This is basically the effective population size. This ratio plus the sample term, yeah? So the sample term is here, and that's and these ones, obviously, they have to be different because the ratio is important. So if this is an estimate that's really close to that one, the effective population size will be really high, and if it's very different, the effective population size will be low. And that's a bit what Robin tried to, this is what Robin exactly says. If the signal is low compared or in the same order of magnitude as your sample kind of noise, you're really in trouble. And it could be even that, because this should be obviously always lower than that. It could be sometimes higher, and you get funky estimates and infinity values for effective population size. So my first check is always, are these estimates different enough from each other so the ratio can be meaningful or not? Obviously, the simulated data, and that works pretty well. Yeah. Okay, and obviously you get estimated in the different versions of confidence intervals. There's a jackknife and a parametric version of that. And I refer probably to the papers to explain that, what they do. But basically different statistic procedures to infer confidence intervals. Okay. Oh, that's great. And you could play around here and do that. I skipped that, but just to let you know, this is now a practical, so you could even run it in here. So you could click here, and then it runs the code you have here, and you can change it, and it should run in the tutorial. So, but because it takes a while, I just skipped that. Okay. Now let's do some corrections for genomic data, because that's the good news. If you have some additional information, we can try to kind of prune our loci even more. So instead of using data on the same chromosome, if you have chromosome information, you can switch on this value, and that means all the loci I put into this simulation are not on the same chromosome. And if you have this information, you basically get an estimate that takes it into account. If you wanted to know, this is an effective population size of 15, and it works really well. And if you run that, maybe let's run it. It's pretty quick. Just make you aware of that. It's kind of a convenient function. This is a GenLite object that has now 2 populations. It's also similar data. That's why it's good. I know the answer. So, sorry. If I just use what I had here, only 100 loci to make it quick, and we have 2 populations. And if you run that, and in this case, we have basically chromosome information, and if you have that, you have to put it into this chromosome vector. I'll show you what it is actually. It's very simple. Here we have this 100 loci, and the first loci is on chromosome 4, the second on 6, 4, and so on. Yeah, it takes it into account, and with this information, it runs it and only uses pairs of loci which are not on the same chromosome. Yeah, so it's one of the corrections you can do. If you have obviously a reference genome, we show later in session 9, if you have a reference genome, you can do something which is called a blast. So you use your dartR object, blast it against the reference genome, and it gives you back from are they the same or not. Obviously, you need a nice reference genome for doing that. And then you get these entries, basically, if you have that. Okay, and we're happy to show that later. And then, obviously, we can run that. And as I said, the answer is actually not really important. Maybe the interesting bit is here, if you have a GenLite object that has 2 populations, it runs it separately for both populations. That's why you get 2 outputs. Yeah. So it's kind of a convenience function, but it assumes that this population are totally independent from each other. So you could have run by 1, you would get exactly the same answer. Yeah. And again, because it's similar data, the filter, minor allele frequency filter, doesn't do much. There was a question, and I try to get there. That's Laura. That's exactly what it is. Yeah. Yeah. And again, if I miss something, obviously this any estimator has a really nice documentation manual. Have a look at there. It explains it in detail how all the jackknives are, all the confidence intervals, all the outputs are created. So it's really an excellent manual. Okay, let's go to the next one. That's cool. Quite often you don't have chromosome data, yeah, but you have something like the number of chromosomes, and that was the slide Robin showed before. You can use a correction factor in the number of chromosomes, and the idea is straightforward. So if you have 50 chromosomes, the chances that you're on the same, if you have a pair of loci next to each other, is lower than if you have only 2 chromosomes. Then the chance you take a pair of loci and they're on the same chromosome, that's pretty high. Yeah. So you can use this information, and Robin found a formula for that. So that's the good news for us. If you know that, you just say, okay, I want to use Waples correction with the numbers of chromosomes, and you have to put in also the value how many chromosomes you have, because that depends the correction factor. There's another setting, just to make you aware, you can also correct for the length of the genome. So instead of Waples correction in chromosome, it is, help me, it is chromosome genome length. Here we are. So if you know your genome length, and then you can use this, and obviously then you have to put in the genome length, and don't ask me about the units. I haven't done that. It's probably in bases, in megabases, gigabases. I have to check. Hopefully it's written somewhere. Yeah. Okay. Yeah, here in genome length in centimorgan. Oof. Okay. Okay. And then you you can run that, and as before, the values here is not really important, but it takes that into account, how that works. I'm just happy that if it runs, so that's why I do it. Okay. Yes, that's probably important. If you run it with your own data, there's quite a good chance that you get infinity values. And the reason for that is, if you do the correction in any version, you basically have a square root, and this one can become negative. So you get an infinity value in your estimate. And the question is that due to sample size or not? You know, if you're low sample size, the chances you get an infinity value is really high. One way to test it, I'm not saying this is the best one. Obviously, you have to think about hard. Maybe my data is not good. Maybe my filter is not good. Any of the things. But one way to test it, you can put in this idea that naive equals true. What is naive? It basically doesn't use the sample correction. Yeah, and that means you run it and you don't get any infinity value. And then you run it without, so naive equals false with the correction, and you get infinity values. Then you have a good chance. Probably the reason why that is due to my low sample size. Yeah, and then you have an idea. It's not because of some funky outliers, bad SNPs, noise data. It's more likely because of my sample size. Unfortunately, it's probably not good news anyway, because it means you have to get more samples. Okay, there was a obviously exercise, and feel free to do that in your leisure and spare time. You know, you simulate different populations, get a feeling for that, and obviously nothing stops you to mimic your data set. You know, maybe you have 30 individuals, you have 500 loci, and obviously they're ideal, and you would see what estimate you would get in an ideal situation, and then can compare your estimate. It's almost like the answer. I mumble a little bit, but Robin had all these caveats we have. Quite often my simple answer is, maybe, help me, it's correct, that you can simulate your simulation as your data set and can try to mimic and put all the information you have in your data of your system in there. You know, maybe there's migration between two populations. You simulate all that and see how much it would affect your estimate of vector population size. And then you get a feeling, I'm not saying this is a good, that's a definite answer, but you get a feeling how much violation of the assumptions can this method, giving my sample size and everything, tolerate. And if it's vastly different and terrible, just depending on the migration rate from 0.01 to 0.2, then probably you wouldn't trust your estimates. If it's pretty stable, then you have a good idea. Yep, that is something. And again, obviously I make that because I have simulation data, a simulation session coming up, and therefore it's really important to do simulations if you can. Yeah, that's good. How much time do you guys have? How do I do that? There's another part coming up, and I basically just outlined the idea, and it's kind of, it's actually my favorite kind of subject. It's a bit unfortunate. It is estimate effective population sizes in a historical sense. So what we can do is we have a beautiful data set, and we can estimate effective population sizes, what happened backwards in time. And this method is called stairways or EPOS, and it has lots of caveats, and I think I'm almost inclined not to show it and do another session and just let you read the manual. Maybe just give you a glimpse into that. How this is run and what it does. So you get an idea of what you can do or what the method does. sec. I knew that would happen. Yeah, okay, thanks. Okay, so I have some sample data, and if you wanted to know, this is a constant population size of the last 400 years of 100. This was a simulation where I started with 100. In 50 years before present, it crashed to 50. This is a simulation that increased. So I started with 100. In 50 years before present, it went up to 200. And this is a simulation where I simulate a bottleneck. It crashed 100 years ago to 10, and then 50 years after that, it recovered to 50. Yeah, so it's kind of very simple scenarios. And when you run this method, you have to learn to look about your something which is called site frequency spectrum. And which is a distribution of alleles. We had that yesterday. And what the method does is basically exploit the distribution of your site frequency spectrum, because the constant one is very different from a decreasing one, not very different. It's subtly different, maybe. If you look at there, you have an excess of alleles in the, yeah, I explained that. Basically, the shape of the site frequency spectrum is depending on the history of the population. And then run all this, I basically estimate now the historic population sizes using that. And it works, as always, beautifully in, hang on, these are just the site frequency spectrum, the 4 of them, you know, in a constant is kind of a reference that are 100. And you can see if you have something like an increase, you have an excess of alleles which have 1 or 2 only occur once or twice in the population. That's the reason, because they have lots of individuals have a mutation rate, so it creates a lot of new alleles. If you have a decline, you have not that many anymore because you lost them. Yeah, drift is really strong. So you can see the different trajectories affect this histogram in a certain way. And obviously there are some people who are really clever who kind of have found a way to exploit that information. Again, we need this one. I like this one. There's another method called stairways. This is one that's really fast and it gives you almost the same settings like the other one would do. There are 2 parameters I explain in a second because that's really important. So if you run that, you can see this is effective population size for the constant simulation of 100, and it gets it. I'm not sure what that was. Am I still visible here? Because my headphone beeped. I'm not sure. Okay. Maybe it means our session is close to end. So you can see this worked actually pretty nicely. You get a really nice effective population of 100. You get some funky artifacts. So for example, this method, that's the first thing you should learn. And we did a simulation. If Isabel is still there, she would do that with an honor student and now PhD student of mine. We did a simulation study with thousands of these trajectories, and we found out this method is pretty crappy in the last 10 generations. So if you use that, don't do that. The reason, obviously, you don't have many information in the last, not many mutations has happened, and there the sampling variance is pretty bad. If I run the same one for the decline scenario, you can see that actually works also quite well. This was crashing 50 years ago, 50 generations ago, be precise. And it kind of mimics also the data quite nicely. So the bottom line, these methods work really nicely with ideal data set. And that's pretty much what Robin said as well. And yes, you have to be really careful to use your own data because they are kind of critical. You have really good sample sizes. You have more or less a closed population. Migration is not a good idea. because it will affect it. And the quality of SNPs you're using should be high, and you need lots of SNPs, and even more important, lots of individuals. And when I say lots, maybe in the order at least of 30, 50, 100 is even better, obviously. In the order of loci, we found out, and we're just about to publish a paper on that, and happy to put up the preprint maybe next week. And there we show we need at least 5,000, maybe 10,000, even 20,000 loci to get reliable results. The method is pretty good to detect a trend. So if you have a declining population, you will get a trend. Don't look too much on the absolute values, because there are two things you have to know. You have to get the length of your chromosomes and the mutation rate. And these are really tricky to get. Mutation rate, you can maybe guess, but length of chromosome is actually not the length of the actual chromosome. In our SNP data is the length of callable SNPs. And you never know that, because it depends how much coverage you have, how much you filter, and all this kind of thing. This is a critical value. Good news, these two parameters don't affect the trajectory. So if you have a decline, you always will find a decline, but it will affect heavily the axis. And not pointing to colleagues, but if you find these methods, there is somewhere in the literature, you sometimes see ridiculous axis values on the X and the Y axis. People look millions of years back and they have effective population size of 1 million, which I find mind-boggling. Why is that? Because just to make you aware, human effective population size is estimated in the order of 10,000 to 20,000. And so really, really, really high effective population values and or generation times is most likely they didn't specify the L value correctly. And just there's some ways to try to avoid that. And I unfortunately don't have much time. But you can use something like the contemporary estimate you get from the LD method, any estimator, as an anchoring point. So if your trajectory ends in the area, and you can rely on your estimate of the effective population size, whatever you have, contemporary, then you have a better chance that the rest of the axis do reflect the actual population size. I'm a bit sorry not to have shown more of that. So it's more a glimpse to make you aware that you can do that with the data. It is, as Robin said, I completely agree on that, tricky, and I would never trust one run and say, this is the trajectory that happened. So you have to be really careful, play with it. The best of the world, you simulate a scenario, get a feeling how stable your answer is. Maybe put in missing data in your samples, or get rid of some of your individuals to subsample and see how stable your trajectory is you find. Never use the first run and say, that I expected a decline, I found a decline, excellent. So please be careful with this one. There is there's another method called Stairways, that's the same thing. And there are also, there's a code provided. And before I stop here, I want to make you aware of the chunks that were provided chunks. It's Robin's terminology, it's not mine. So he provided chunks of code that really nicely illustrates some of the principles, and it's, as he said, very simple R code, and I like them a lot. They should run. So for example, there's one which I will show in the simulation session, and good news is I don't have to do because I do it now. It's a simple simulation which is beautiful. For example, here, you start with an allele frequency of 0.5 and have a certain population size. And this basically shows what happens to this loci if you simulate it forward in time in terms of drift. And if you would kind of have a lower number here for your generation time, for generation size, that would go to fixation earlier. And if you have a high number, it's lower. So basically, these are really nice little chunks that gives you the idea of all the methods, and you can play with it. And I strongly recommend, to use them if you have some estimate of your effective population size, and you can then try to validate that in using this kind of little code snippets. Are the values I see, do they make sense and do they align? For example, I think this one is a full version of the NE estimator in terms of, obviously with lots of simplifications, but what you can get out of in terms of effective population size. Yeah, so very similar to the one where I simulate my SNP data, but this is done on kind of, yeah, it's actually the same thing. It's just not estimating SNPs, it's run on allele frequencies. Okay. I think that's it from my side. Any questions? I think there are some, and then I would stop because we have another session coming up and we're getting into territory of the next one. Is there any use the blast feature against a good reference genome? Does it have to be with the reference genome or the same? You know what you estimate is, are these two loci on the same chromosome or not? So if you have a close species and the numbers of chromosomes is the same and there hasn't been much rearrangement, yes, probably a reference genome is fine. And obviously, the further away the species, the less loci you will get back from your blast because something happened in terms of mutations or anything. So yes, in principle, you can do that, but you would like to have some indication this species you're using for a reference genome is very, very similar to the one you're having. And what is very, very similar? I don't know. Hope that makes sense. Any other question? I don't have the chromosome information, therefore I don't have the linkage distribution between loci. Is it still possible to calculate? Yes, it is, but be aware you get into this caveat that some of your loci will be linked, and that creates noise and distortion and bias in your estimates. Brief, isn't it? It's not possible. Okay, my answer is I can show you in a paper we kind of close to publish. We did that. We had estimated the NE to NC ratio. So we have capture recapture of the population, and we estimate NE using the linkage disequilibrium method. And we can show if you have less than 30 samples, your estimate is all over the shop. It doesn't work very well. So it's almost unfortunate, but I would treat that estimate with very, very caution. Yeah, obviously you get an estimate, and if you have a highly endangered species and your estimate is 50, then that's probably reasonable. If you get 5,000 and you say, this population is safe, but obviously this is confirmation bias, so I don't have a good answer for that. You know what you could do? You could try to run subsamples, even from your small sample size, and if you get similar outputs, you get maybe some confidence that you have data, or you filter harder and less hard. And if it's a stable, you always get similar estimates. I would start to believe it's okay.
Right.
Yep.
Yes, maybe you can stay. You don't have examples.
Yeah, it's true. It's true that, on the plus side, and Chris just reminded me on that, is also what Robin said, when you have a low population size, drift is strong, so the signal is strong, so it works in your favor. So maybe you can get away with 20 samples because you really have a highly endangered species and your R-square value. Please look at that ratio I just showed you, the R-square value of the sample and the R-square value from your estimate. If they are fairly different from each other, you have a better chance that you get actually a real signal and not just noise. Okay. Is it safe to go another minute or two to answer some of the questions, or you wanna? These are answered. Missing data and samples. No, don't. Impute is a tricky one. Maybe is Robin still there? He would tell me. You know, if you can impute meaningful, that would be nice. But quite often, it's better to avoid missing data. So you should filter for loci that have lots of missing data. You know, if you have only in R, if you filter for call rate of 0.95 or so something, that has not many missing data. I think that should be okay. That would be my answer. Make it as small as possible, and rather better than have less loci of high quality.
Bernd, can you hear me?
Sorry?
Yeah, this is Robin. Can you hear me?
I see.
Okay, yeah. I would not try to impute.
That's why it was beeping, is it?
Missing data for any. There's no problem in missing data as long as it's randomly missing. But if it's missing because of something related to the actual genotypes, that's a worry.
Thanks a lot. It makes sense. This is okay. This is just for me to using a subset. That's basically just a population definition. I explained that if you want again somewhere else. Okay, last one. Isabel, why do you do that? L bias. As I say, it's not critical if you're just interested in the trajectory. The best way, obviously, is you would have an idea what is the size of my chromosomes, of my genome, and then check if your L is in the order of magnitude. Makes sense. So if you have high coverage and your L is larger than actually the genome size, then you have trouble. So you can kind of get an idea. Normally, it's something in the 10% of the length of the chromosomes, but it really depends on the coverage and your filter. So I'm careful with that. But you can get some idea what is a sensible L in your data. It wasn't a good answer, doesn't it?